Good morning, dear friends. We are again back here with the point called as sterilization and disinfection. This is the part for BSc first year microbiology students. So now let us understand these certain terminologies, because these terminologies will be helpful for us in understanding the basics of how an infection takes place and how microorganisms are being killed because of certain methods in microbiology which are helpful for us in getting pure culture. Last lecture was mine on pure culture, different techniques we have seen, and the sterilization and disinfection parts comes thereafter. Now what we are going to see here, we will directly go to the content, and the main part that comes in play, it is called as sterilization. So what is sterilization? Process by which an article, surface and medium is freed of all microorganisms either in vegetative or spore state. Means whenever sterilization, the word sterilization comes in your mind, it is being seen that this process is freed of all microorganisms. By sterilization, we are making any sort of thing to be without microorganisms. Therefore, it should be freed from microorganisms. It may be an article, it may be a surface, it may be the media that is being utilized for microbiological growth also, which is to be sterilized. So, on that basis, the organisms are to be freed, means it has to be removed and it has to be destroyed, either in vegetative or spore formed state. So, that doesn't make a difference at all. But sterilization will be a process where there will be complete removal of microorganisms. Now another term that comes in play, it is called as disinfection. It means destruction of all pathogenic microorganisms or organisms capable of giving rise to infection. Okay. So these terminologies are there, wherein there are certain little bit amount of confusion. Destruction of pathogenic microorganisms. Whenever there is pathogens or pathogenic microorganisms are there, then and only then the destruction would take place, and the organisms which are capable of getting infection, those organisms when being removed, it is called as, the term called as disinfection. Now what is a disinfectant? A chemical used or non-vital object to kill surface vegetative pathogenic organisms, but not necessarily spore formers or viruses. Now in case of sterilization, we have seen that whether it is in the vegetative form or in the spore form, the killing of microorganisms would be taking place. But here, whenever disinfectant, when we are utilizing in case of the term disinfection, then it will be helpful for surface vegetative pathogenic organisms. It will be only killing specific amount or specific type of organisms. Basically, this vegetative form of our microorganisms are being used, while this spore form or viruses cannot be destroyed because of the disinfectant or the process of disinfection. Antiseptic. Now whenever there is a wound or something, we apply antiseptic. So what is an antiseptic? A chemical that is applied to living tissues such as skin or mucosal membrane to reduce the number of microorganisms present through inhibition of their activity of destruction. So if I get a cut over here itself, so what I put in over here is the disinfectant. The most important part that comes in play is disinfectant. So once we put in disinfectant, because of the disinfectant, it will only have surface area which will be covered, or it will penetrate inside where the mucous membrane, and ultimately it tries to kill those organisms which are present over there and reduce the microorganisms so that it won't be causing any further infections. Now method of sterilization, two basic methods are been there. Physical agents are being used and chemical agents are being used. So there are certain physical factors by which one can go along with the process of sterilization can take place, or there are chemical sterilant also by which the process of sterilization can take place. Let us understand one by one. Physical agents. Now what are the physical agents that are there? Sunlight, drying, dry heat, moist heat, filtration, radiation, ultrasonic and sonic vibration. So these are various different aspects or terms on the basis of which we can understand the process of sterilization with the help of physical agents. Now let us see. Dry heat. Mechanism of action. Protein denaturation, oxidative damages, toxic effect of elevated level of electrolytes. So by dry heat also we can easily calculate or we can easily see that the process would take place. Now how this process would take place? It may be taking place by different mechanisms. Protein denaturations, the proteins which are present on the surface of that particular bacteria may be destroyed. Oxidative damage, the process of oxidation reduction reaction may be destroyed and therefore the whole mechanism would collapse. Toxic effects of elevated levels of electrolytes, excess amount of electrolytes are been there, then also because of dry heat the sterilization would take place. Now what can be done with the help of this dry heat? How do you apply dry heat? By flaming, incinerator or hot air oven. Flaming or incinerator or hot air oven. Let us understand. Flaming. The articles are passed into the Bunsen burner flame. Articles are made red hot. You can see here. This is the Bunsen burner that you can easily see. And if you see the blue flame of the burner, this is the most sterile area, wherein if you put the nichrome wire loop and any organisms which are present in the nichrome wire loop will be get destroyed. This is called as pure flaming. Article sterilization, incubating loops of wires, forceps, spatulas, mouths of culture tubes. So all these things are to be utilized for making up that media or for transferring of the pure cultures. There and therefore it has to be pre-sterilized. So for the sterilization method, flaming is one of the best methods that one can work on. Contaminated material in bulk is sterilized and disposed by burning in an incinerator. If you just observe this figure, this is then incinerator. Incinerator is a kind of thing where it is in a kind of chamber. If you put in something and if you start it, then it on both the sides it will directly start with flame and everything will be burnt out. So articles which are sterilized: surgical dressings, disposable syringes, contaminated lab materials, animal carriers and beddings also can be destroyed, because these all are said to be a source of microorganisms. Generally the hospitals utilizes such kind of incinerators in order to destroy those particular used type of gloves or beddings or some kind of disposable syringes basically which are directly now been thrown. These can also be put into the incinerator and the incinerator can burn it out. Now, Louis Pasteur discovered in 1986 the compartments with the perforated trays and fans. The temperature is 160 degrees for 1 hour. Preserve sharp edges of cutting instrument. This is the hot air oven. If you observe this is the hot air oven. It has different trays inside it, and onto the trays you can keep your equipments and something, and it has been what we can say. If you put it in 160 degrees centigrade for 1 hour, then the proper level of sterilization can be achieved. See this. So what articles can be sterilized inside an hot air oven? Scissors, scalpels, glass syringes, swabs, liquid paraffins, testing powder, fats and greases, glassware and forceps. So these can be put in. You can just observe this. This can be put in inside those particular hot air oven, and once you put the lid of that particular hot air oven and you start the hot air oven for 160 degrees centigrade for 1 hour, then you will be able to sterilize all these instruments or the lab equipments very correctly. Now, what sort of precaution has to be taken when we are using a hot air oven? Temperature should not exceed 180 degrees because glassware kept inside for sterilization will get smoky. Appearance and wire paper used to cover the articles will get charred, and the glassware kept inside should be totally dry or they will be breaking. Okay. No sudden cooling of hot air oven, no overloading of hot air oven should take place. Because if hot air oven is cooled suddenly, then again there will be destructions of that particular hot air oven as well. Sterilization control. Brown's tubes are generally utilized. If my sterilizer or if my hot air oven is working in a proper condition, it's absolutely fine. But if it is not working in a proper conditions, or for example, if I bring from the market a very brand new hot air oven, but I don't know whether that hot air oven really sterilizes it or not, then what you can do, you can perform a test which is called as Brown's tube. This is been utilized for proper sterilization. Green color indicates the proper sterilization. Non-toxic strains of Clostridium tetani Spore germination indicates improper sterilization. So these are certain indicators by which we can understand that the organisms are not present there, or the working of that hot air oven is done in a very correct manner. Therefore, we can use this. For example, Brown's tube can be used. This Brown's tube, it contains a liquid which is green in color and which is transparent, and if after sterilization cycle, if it turns green, then we can say this proper process of sterilization is very proper and our instrument is correctly being monitored. Second part, a microorganism strain of Clostridium tetani, non-toxic Clostridium tetani is being utilized. You can just put it onto the plate and put it inside the hot air oven. After the incubation, if the germination of spore takes place, it means that the hot air oven is not working in a proper manner. Glass bead sterilization employs a heat transfer device, glass beads and salts. Article can be sterilized, endocytic files and birds. Temperature 220, temperature 10 seconds. This is not been utilized in a very daily format. Moisture. Now this is most important. Mechanism of action, how moisture takes action, mode of action of moisture, denaturation of proteins and coagulation of proteins will take place because of this particular kind of thing. Temperature below 100 degrees and temperature at 100 degrees will be helpful for proper sterilization. Pasteurization. Now this is most important. Holders process, this is called as 60 degrees centigrade for 30 minutes. You hold that particular content up to 63 degrees centigrade for 30 minutes only, and you will get a proper way of sterilization. Flash process, 72 degrees centigrade for 50 to 20 seconds. This will be helpful for killing all those microorganisms which are present, and the process is called as pasteurization. Destroys Mycobacterium, Salmonella and also Brucella, Klebsiella burnetii survive older methods. So these, because of this particular kind of, if you just observe, this is the what we can say. This is the milk industry, wherein the milk goes through such kind of chambers. When the milk has been pasteurized, because of this particular process, the level of contamination gets reduced down. Temperature 100 degrees, boiling. It's one of the most important way by which we can sterilize. Vegetable bacteria killed at 90 degrees. Time required is 10 to 30 minutes. No effective for spore bacteria. Sterilization promoted use of 2% sodium bicarbonate. So even by what we can say, boiling water also sterilization would take place, but it doesn't kill the spore form of the particular bacteria, while the vegetative forms can be easily killed out. Sterilization for media containing sugar or gelatin, exposure 100 degrees for 20 minutes on successive 3 days will be helpful for sterilization. Because sugar, if you what we can say, sterilize sugar in a very normal way, or by heating it, charring of sugar would take place. Some brownish color would appear on the sugar, and therefore the sugar may get damaged, because some media are there, bacteriological media, which contains sugars, and therefore such kind of media cannot be directly sterilized into a what we can say, into a sterilizer, and therefore such kind of sterilization method can be employed. Steam under pressure. This is autoclave principle. Water boils with pressure equals to the surrounding atmosphere. Saturated steam has penetrating power. In downward displacement, air in the chamber is forced downwards and out of the bottom displacement. Pre-vacuum high temperature, most modern, economical. See, autoclave time. This time is said to be most important time. 121 degrees centigrade. Pressure should be 1 lbs and for 15 minutes. So 15 to 121 degree, 15 and 15. If you proceed to 126, 15 and 10 minutes, and 134, 15 and 3 minutes also. So this temperature can be helpful for the autoclave to work out very efficiently. Articles sterilized, surgical instrument, lab equipments, metallic syringes, all culture media except media containing sugar and gelatin can be sterilized into this particular kind of thing. Sterilization control, how we can check. The best possible option is Bacillus thermophilus. This is the spore of the Bacillus thermophilus. Autoclave tapes are now been available, color changing tapes, which you put inside and you can easily go along with. Agents to avoid corrosive reaction of the steam, ammonia, 2% sodium nitrite and dicyclohexaamino nitrate are being utilized. Okay, fine. So we'll stop here by today. In my next lecture we will continue because that may create confusion. So what we have learned, we have learned two methods today. One is called as the direct method, direct because of the hot air oven or sterilization we have seen, and second is moist heat. One is dry heat, one is moist heat. We have seen two these kind of methods. In my next lecture we will try to continue with what is radiations. Okay, thank you.